Hello guys, I hope you're all doing well and I hope you're ready for chapter 7 and 11. They both talk about membrane and so I put them together. I think we talk about them together. So anyhow, let's go on and talk about what are the components of chapter 7. So it's about membrane, cell membrane. As I said, before, I'm going to bring you some Oreo cookies. Of course, if we were in a classroom set, we would eat them together. Let me pick up the thickest one there. I don't know, I got these from a different store than the normal usual. Oh, here is a good one. Yes, can you guys see this? Okay, right in between the two layers is a cream. So that's what cell membrane look like. I hope you guys can see that. Anyhow, I'm not gonna eat it. I'll wait. There's not much left of it. My family, they're like vultures. I buy them for myself and my family crave on them. Anyhow, so a cell membrane, the plasma membrane, another name for it, is a boundary of the cell that separates inside of the cell. I better get my cells out. Those two cells that I showed you before. I better hang on. Get them out. So, oh, come on. Yeah, not okay. So, I hope. So they are boundary of the cell, and the plasma membrane it shows a selective permeability. It means it chooses what can go inside of the cell or it cannot go inside of the cell. They have a brain. Well, they do not have a brain, you know that. But they are selective as far as what they do, okay? Anyhow, what is the structure of cellular membrane? A fluid mosaic model, and we need to talk about that. Phospholipid, we talked about it, that it's a two fatty acids here, and the head is a it's a glycerol and phosphate. Remember that. So it's an amphipathic. That's a good term that you should know. Amphi, it means the two lives, like amphibians. You've heard of the animals, amphibians. They have two lives. So these molecules, these phospholipids, they are two ends. One end is hydrophobic, and the other end is hydrophilic. It loves water, this end, and this end does not like water, okay? So it's an amphipathic containing hydrophilic and hydrophobic regions. A phospholipid bilayer can exist as a stable boundary between two aqueous, as I said, outside of the cell. This is a cell, outside of it is aqueous, water, and of course, inside of it is aqueous as well. The fluid mosaic model, I will talk about, well, let me give you the definition of fluid mosaic model now, and then later on, I will elaborate on it. The fluid mosaic model, it says, for example, if this is a phospholipid bilayer, these white structures you see, they are protein molecules embedded in here. Those white structures in the phospholipid bilayer, they do move around. They're not stationary. Like if the door to your house is stationary, you cannot move the door to your house and put it in the garage door. Okay, I hope near the garage. Where it stays is static. It's not fluid. Okay. But you are fluid. You come in your house and you get out of the house, you are fluid. So the cell membrane is fluid. I think as time goes on, we will talk about it. So my Oreo cookies right here, not much cream in here. Which one was that cream? Right here, phospholipid. So up here is the phosphate head, and the hydrophobic tails. Okay, these are hydrophobic tails, and then of course outside of the cell is water. As you can see, inside of the cell is also water. Okay, we talked about this a little bit in the past, and this is putting everything together for you guys. That's okay. We'll talk about it. Let's talk about it. And then these are, if I would have brought some chocolate chips and embed them in here, you guys would know what I'm talking about. I'll bring some chocolate chips and embed them here. That would be these protein molecules. That would be those protein molecules being embedded right into this Oreo cookies. Okay, so then attached to these protein molecules are some carbohydrates. Some of these protein molecules, not all of them. Some of these protein molecules that they are called glycoprotein, the combination of that protein molecule and that oligosaccharide is glycoproteins. And then those oligosaccharides can attach to the fats. It's called glycolipid. So those are the terminologies I would like you to know. They're significant, they're important. Then one more thing, last thing that I need to mention about cell membrane, there are cholesterol embedded within the cell membrane, okay? So there are cholesterol in there. You can see them here, you can see it here, you can see it here, you can see it here and here. So these cholesterols make the cell membrane, we'll talk about it will come up. So what happens outside of the cell membrane, extracellular matrix, remember we talked about that, that's outside of the cell membrane right here. And of course, in case of cancer, they can be a problem, too much excess of it. But in most cases, they are like a protective layer outside of the cell. And then inside of the cell, the microtubules, microfilaments, we talked about this a little bit before. There are 3 types of cytoskeleton, and I talked about it in the past exam. And they are inside of the cell, hold up the cell together. And the protein molecules, they call them integral proteins molecule, peripheral protein molecules. They're inside of the cell, okay? So they are called peripheral protein molecules. And I think I talked about everything. We'll come back and revisit most of these things again, guys, okay? So, anyhow, let's move on. Then fiber of extracellular matrix, we talked about this a little bit. Glycolipid, glycoproteins, it's been talked, okay. What is fluidity of membrane? Phospholipid, as I said, even the phospholipid within the cell membrane, they're fluid as well. Not just the proteins. The phospholipid in the plasma membrane can move within the bilayer, okay? So those phospholipid molecules, they do move around within the phospholipid bilayer. Most of the lipid and some proteins drift laterally. So the protein molecules can drift, go in a lateral way, and rarely the phospholipids flip-flop, okay? It can do that as well. They call them a flip-flop transversely across the membrane. Okay, here is, based on this experiment early on, scientists found out, and they do it every day in laboratory nowadays, they found out that the cell membrane is fluid, okay? It's not static. So they took a mouse cell and they took a human cell. Of course, they're showing you different proteins, protein molecules on the surface of the human cells, and the mouse cells, they're showing them in pink for the mouse and blue for human. And when they combined the 2 cells, they hybrid them, they found both proteins mix after 1 hour. And then, as I said, they do this every day in laboratory for monoclonal, making monoclonal antibodies. Okay, what are properties of membrane? Okay, let's go over that. A temperature cool membrane switch from a fluid state to a solid state. When, of course, just like oil, you take a vegetable oil and you put it in a cooler or you put it in a freezer, they freeze, okay? So the temperature at which a membrane solidifies depends on the type of the lipid. The membrane rich in unsaturated fatty acid, very important concept. Lipids that are unsaturated fatty acids are more fluid than those in the saturated fatty acids, okay? And remember, we talked about it in the past that usually the animal fat are saturated fat. These are animal fat, saturated fat. And plant fat are unsaturated fat, okay? And put those two concepts here together for yourself. Membrane must be fluid to work properly. So they are usually about as fluid as a salad oil. And then they're asymmetrical. It means the Oreo cookie that I showed you pretty much is symmetry. But if I have a chocolate chip embedded here and a chocolate chip embedded here, it means that side and this side are not symmetry. They are not exactly alike. This is symmetry right now. This Oreo cookie is symmetry, okay? But cell membranes are not symmetry, okay? If I go back a few slides, it will come up. You will see that the protein molecules on this side are not the exact same thing on that side, okay? So these are all properties of cell membrane. The steroid cholesterol has different effects on membrane fluidity at different temperatures. At warm temperature, such as our body temperature, 37 Celsius, cholesterol restraint confirm the movement of phospholipids, okay? So cholesterol in a way is good that the phospholipids do not move around too much. It anchors them down. How about that? At warm temperature, around 37 Celsius, which is our body temperature. At cool temperature, it maintains fluidity, but preventing tight packing of the cell. And membrane is built by endoplasmic reticulum and Golgi apparatus, which I talked about it in the last chapter a little bit. Here they are. They're showing you. I better put my glasses and make sure that I know what they are saying. Unsaturated fatty acids, unsaturated viruses versus saturated hydrocarbons. Here is a saturated, very viscous, very tough type of cell membrane, very tight. And here's a fluid type of membrane. You have a lot of unsaturated fatty acids. And then when you have cholesterol in there, it reduces membrane fluidity, okay? It's not gonna be as fluid as this membrane, okay? And it's not gonna be as tight as this membrane, okay? So membrane fluidity at moderate temperature, but low temperature hinders solidifications, as I talked about that. Okay, what are membranes, proteins and their functions? We have peripheral proteins. I showed you they're inside of the cell membrane. We have integral protein molecules. They are embedded within this is phospholipid. They have protein molecules embedded within in there. So anyhow, and I showed it to you. Integral proteins that span the membrane are called transmembrane proteins. And I got to show you some pictures. They are a type of integral proteins. Transmembrane, they're different type of transmembrane, integral membranes, and then transmembrane is one type of them. The hydrophobic regions of an integral protein consist of one or more stretches of non-polar amino acids. Remember, I talked about, you guys, you should know the polar and non-polar amino acids from last exam. Now this exam, you do need to know that. And often coiled into alpha helix. Here they are. So here is a transmembrane inside of the cell, outside of the cell. Here is the protein molecule N-terminus. In a classroom, I would ask you, what is N-terminus? That's where amino group is, okay? And this is the C-terminus right here, the other end of the amino acid. Remember amino acids, you remember that, okay? So one end is C-terminus and the other end is N-terminus. Okay, extracellular side, cytoplasmic side, and that's a integral proteins. It's not a peripheral protein. How do I know? Because it's embedded within the phospholipid protein. 6 major functions of the membrane proteins. Yes, you definitely need to know these, and then we'll talk about them for the rest of semester, at least for this 2 chapters, and chapter 11 comes into picture, and these are very, very important. It transports, of course, the protein molecules are designed to transport molecules from outside of the cell to inside of the cell, or from inside of the cell transports molecules to the outside of the cells. Enzyme activity, they act like enzymes, and you will see that, cyclic AMP. The signal transduction, so they are they recognize some of the molecules, and then of course they open up, they allow molecules go inside of the cells, and you will see that they're like a signal. The protein molecules act like a signal on the surface of the cell membrane. Cell-to-cell recognition, 2 cells recognize each other. That's chapter 11 material. Okay, these 2 cells recognize each other. Okay, and then of course they exchange materials or what have you. Okay, I had another one. These 2 cells recognize each other. Intercellular joining, yes, there are 2 cells can be combined together, and there, last exam I talked about, or last chapter, you know, gap junctions and desmosomes. I gave you a whole long list. Okay, and then of course attachment to cytoskeletons and extracellular matrix. I think that the E and F, they kind of go hand to hand. Here they are, they're talking about it. So right here, transport, when ATP attaches to this, the molecules can get in and get out. It's a transporter. Enzymatic, they, and I will talk about them. They're showing you all of these. I'm gonna go ahead and cell-to-cell mediated, and then intercellular, and then of course attachment. We'll talk about this. HIV, so there are protein molecules on the surface of our white blood cells, right here. These protein molecules on CD4 are white blood cells, CD4, CD8. They have these receptors for HIV, and when HIV attaches to them, of course, the HIV can get inside of the cell. For example, your liver cells do not have these receptors, okay? As I said, all of the 200 some odd cells we have in our body, different type of cells, the receptors on them is different. Just like this cell and that cell, they're different. The white, which is a receptor, is different than the yellow and so on and so forth on this cell. Here is a white blood cell, here is a liver cells. Okay, they have different receptors. Okay, I think we did talk about this. Membrane is built by the endoplasmic reticulum and Golgi apparatus. We talked about this. So when the membrane is built and they transport molecules outside of the cell, so that vesicle, if you would, these vesicles, they attach here to the cell membrane, right here, and they do not pinch off and go back. Okay, that's the current theory. The current theory is that these vesicles come and they become, this becomes part of the cell membrane right here. I hope I'm making some sense. It becomes and become part of cell membrane. It's not like they dump their stuff, exocytosis. You remember that? And then they pinch off and they go back. That's not the case. So hydrophobic, did I talk about everything here? Attached carbohydrates, yes, we talked about this, the carbohydrate, I talked about the last exam. Make sure you study that, glycolipids. So anyhow, hydrophobic nonpolar molecules such as hydrocarbons can dissolve in the lipid bilayer and pass through the membrane rapidly. So these are nonpolar molecules, can go through very rapidly. And hydrophilic molecules, including ions and polar molecules, do not cross the membrane that easily. Okay, so ions and polar molecules do not go through easily, but hydrophobic non-polar molecules, they can go a little bit faster. Of course, this is a general rule. it's not true about everything. For example, water molecule is a polar molecule and it can go through the membrane easier than others. Of course, there are gates, there are openings, aquaporin for water molecules to get in and get out, but they're easier to go in and get out. What are transport proteins? Transport proteins allow a passage of hydrophilic substances across the membrane. Channel proteins like aquaproteins, I talked about that. Aquaporins, I talked about that. Other transport proteins called carrier proteins and bind to molecules and change shape to shuttle them across the membrane. A transport protein is specific for substances it moves, of course. All of the proteins are specific. What is passive transport? Passive transport is when molecules go in and outside of the cell without any ATP needed. Okay, the definition, maybe this is a good place to give you the definition of diffusion. Passive transport and diffusion, they are two different things. Passive transport is when molecules go from high concentration to low concentration through a membrane without ATP needed. But the definition of diffusion, I gave it to you in the lab. When the molecules go from high concentration, if you have a beaker of water and you have some sugar molecules here, these sugar molecules will disperse after 30 minutes or so, will disperse through that beaker of water. Okay, that's diffusion. When you have a bottle of a cologne and you bring it to a room, it diffuses throughout the room and there is no membrane involved. There is no membrane here involved in that sugar solution in that water or salt solution. That is diffusion. That is the definition of diffusion. But if you have the same concept and then you have a membrane, you have a cell that is membrane involved in here, then, and there is no ATP involved, when you want to dissolve that sugar in that beaker of water, there is no ATP involved. You just sit it, you just let the sugar sit in here, okay? That's it. It will be dissolved eventually. You can spend some energy, spin it or shake it, heat it, so it will be dissolved faster, but eventually it will be dissolved in there all at once, okay? But passive transport is the same concept when there is a membrane involved. Although each molecule moves randomly, diffusion of population of molecules may be direction. Okay, here is talking about molecules of a dye going across a section of membrane and so on and so forth. I'm not gonna animation, you know, let's see what this one says.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
Very good. So everything I said, I put it in perspective and a nice video. You remember, easy way to remember it, you guys, okay? So if I have some drops of fat on top of this water, eventually they all come together, okay? So think about it this way. Phospholipid is lipid. So lipid molecules that are not polar, they go easily through. But something that is polar cannot easily go through, okay? So, I don't know, maybe it's something to remember it. What is passive transport? Substances diffuse through the concentration gradient from high, what I mean by concentration gradient, from high concentration to low concentration. The example of the bottle of perfume, the example of bring sugar or salt and put it in this bottle of glass of water. Region along where density of the chemical substances increase or decrease. No ATP needed. When we're talking about passive transport, I already talked about that. And remember, ATP adenosine triphosphate. You break one of the phosphates, it gives energy. Okay, the diffusion of a substance across a biological membrane is passive transport because no energy experienced in the cell. Already talked about all of that. Osmosis, on the other hand, osmosis, you guys, it is the definition of osmosis. Of course, the book gave you another definition, same thing. The definition I'm giving you right now and the definition that the textbook gave you is the same thing. So osmosis says when water molecules go from high concentration to through a low concentration through a selective permeable membrane. That's the definition of osmosis. So you have water molecules, you have a whole bunch of them here, and you only have a few here. So the water molecules are going from high concentration to a low concentration through a selective permeable membrane. That is osmosis. And of course, no ATP is needed. Okay. The definition your textbook gave you, same thing. Water diffuses across a membrane from region of a lower solute. Okay. So when there is more water here, what does that mean? It means there is less solute. I'm going to draw solute as a little squares. There is less solute here. Here, there is more solute. Okay, like salt, sugar. So what happens to the water molecules? They go from high concentration to low concentration. So everything depends on the solute. Two things are involved, solute and water. The definition says the water molecules move. The solute do not move. Okay, so the solute are not moving. Let's assume that for a second. Okay, again, cells are different, but in the lab, I had dialysis bag for you guys, and only water moves. Okay. Molecules can move as well. But anyhow, so that's the definition of osmosis. When water molecules go from high concentration to low concentration, of course, your textbooks elaborate on it, where lower solute concentration to region of a higher solute concentration until the solute concentration is equal on both sides. Here it is. That's what they're showing you when it's an experiment. I'll let you guys look at it. Again, I don't want to spend too much time, but if you guys want to understand the material, you better look at these pictures and try to understand them. It will give you a better understanding. Tenacity, it means strength. Okay, so ability of the surrounding solution causes a cell to gain or lose water. Isotonic, it means both sides, and I'll show you some pictures, both sides of the cell, inside of the cell and outside of the cell, has the same solute. Okay. Hypertonic solution, it means outside of the cell, solution outside of the cell has more solute than inside of the cell. Hypotonic is when inside of the cell, the outside of the cell, it's still talking about outside the cell, has less solute. Okay, so maybe I should write these things down, and then you guys, hypertonic, outside, and we're always talking outside has more solute. Outside has less solute. Solute is sugar, you already know. Okay, very good. Let's look at it. So in case of animals, they call it lysed and normal isotonic solution, inside and outside are same. And they call it, in case of animal, what they, or a shriveled, these are the, in plants, they call it turgid and normal, and they call it plasmolyzed. So when there is a hypotonic, there is less solute outside of the cell, inside of the cell is hypertonic. Okay, so what happens hypotonic outside of the cell, there is a lot of water goes inside of the cell. When there is a lot of water going inside of the cell dies. Hypertonic, the water in the ocean, when you've heard of that expression, water everywhere, not a drop to drink, right here, we're talking about that. So there is a lot of salt in the ocean water. When you stand it in the ocean, do not drink the water. Your cells will shrink. Same thing in plants. And then you are using these experiments. Very good. This is something great. I want to show you guys this because this is something we do in the lab. I've done it in the lab for you guys, but here they are showing the video of it. I showed you the pictures of it. So great. This is a plant. This is an aquatic plant, which is called Elodea. I don't know if I can write on this. Elodea. That's the name of the plant. Elodea. That's an O. And what happens, you will see here that cytoplasmic streaming occur. These chloroplasts, these little dots, the green dots are chloroplasts. And then these chloroplasts, they do move around. They go around the cell, okay? And then watch what happens. I guess you don't have the volume. And then, oh, I guess I have to talk. I narrated. And then they add salt solution to it, and look, the cells are shrinking. This is what we did in the lab, and then that's what you guys used to watch on the microscope. And all of that cells shrinking, they stay in the middle of the cell is shrinking, but the cell wall, these are all cell wall, okay? The black, this is the cell. Right here is a cell that is shrunk. Okay, so again, plasmolysis, the name of the process is plasmolysis. And then of course, we're showing you the osmosis, movement of water molecules from higher concentration to low concentration. Water molecules have a higher concentration inside of the cell, so they leave inside of the cell. When they leave inside of the cell, water molecules, of course, what happens to the cell? They shrink, they shrink, they shrink, they shrink. Okay? And then you can see the cell wall. Target, let's see. I guess, am I the volume? I guess these do not have volume. Okay, same thing. This is showing you the cytoplasmic streaming. You saw it in the last chapter. I talked about it, cytoplasmic streaming. And then when there is too much, the environment is hypotonic, what happens? It disrupts the cells from the normal. Of course, they have to focus the microscope for this experiment. Okay, water balance of cell with cell wall. Cell walls help maintain water balance. Of course, plant cells, hypotonic and target flaccid. We talked about all of this. I'm not going to go ahead and focus on plants too much. But again, make sure you study these and you know that. What is facilitated diffusion? Passive transports aided by proteins. In facilitated diffusion, transport proteins include channel proteins and carrier proteins. Echo proteins facilitated the diffusion of water. Ion channels facilitate the diffusion of ions. It means the protein molecules within the cell membrane, they open and close, so the ions can go in. And you will see an example of it here in a minute. Some ion channels called gated channels, open or close in response to stimulus. Right here, you can see these channels. Of course, they are not mentioning any ATP involved here. So the ions are flowing in and flowing out, in and outside of the cell. Okay, what is active transport? So passive transport, no ATP needed. Active transport, you do need, Houston, we need ATP. Have you watched Apollo 13? Anyhow, I always say that. So in active transports, it means there is energy needed and move solutes against their gradient. It means there is a high concentration here, lower concentration here, and what happens, it moves these guys to that side against their gradients. Okay, facilitated diffusion is still passive because the solute moves down its concentration gradient and the transport requires no energy. We already settled that down. Now, active transport, solutes move against their concentration gradient, okay? And of course, ATP is needed. You need it. It's just like flood comes, okay? And flood is in the street or everywhere. And you got some water inside of your house. You want to pump that water from your house to the street, okay? So you need to get a pump and you need to pay PG&E. You're going to spend energy to get this water out. But the water that comes to your house, no energy needed, just come in. Okay, that would be facilitated diffusion. The water that is coming from outside to your home, God forbid. Okay, but the water that you need to pump out, of course, you need to spend energy, and that's paying PG&E. Specific proteins embedded in a, okay. One example that you should know is sodium potassium pump. The rest of your career in biology, you come across this sodium potassium pump is one type of active transport system. And what happens, active transports allow cells to maintain concentration gradient that differ from their surroundings. A sodium potassium pump is one type of active transport. And what happens with sodium potassium pump, again, I'm not talking, there are protein molecules involved here, and 3 sodium goes outside of the cell, and 2 potassium comes inside of the cell. And eventually the potassium diffuses out through channels to the outside of the cell. So what happens in this case, in case of your neurons, and of course there is 1 ATP needed for this. What happens when 3 sodium goes out and 2 potassium coming inside of the cell, there is 1 ATP spent, and this channel, this protein molecule here, they showed you all of the oligosaccharides attached to protein molecules, the different type of A and B protein molecules. So what happens, 1 ATP needed, 3 sodium comes out. So in a neuron, in a nerve cell, you have a lot of sodium outside of the cell. And then what happens when there is a stimulus, okay, there's a poke, then all of the sodium comes inside of the cell. So you can feel the poke. Yeah. That is totally for next chapter, next course, I'm sorry. I will talk about action potential in zoology bio 2. Okay, passive transport and active transport. You can see the differences. The diffusion, molecules are diffusing through the membrane, and then facilitate diffusion, there's still protein molecules involved. The difference is, like this guy, ATP is not needed. Active transport, molecules go in and out, it depends what you're talking about, but ATP is needed. Okay, membrane potential, the voltage difference across a membrane voltage is created by differences in it. I talked about that a little bit when there's sodium outside, great amount of sodium outside, then outside of the cell is positively charged, inside of the cell is negatively charged. This is a cell membrane, okay? And of course, well, here is a cell membrane, put it this way. I think I've been showing you cell like this. So when you have a lot of sodium outside, when we talk about axon, and all of these sodium, you learned that from first exam material, they're all positive. So can I just write down positive here? And inside of the cell is negative. So what happens, this is called there is a potential. If I attach an electrode, one electrode outside and the other electrode inside, and attach this to the voltmeter, and the voltmeter is showing me it is inside of the cell is minus 40 or 70, minus 70 millivolt. Very small voltage, but still a 70 millivolt. Okay, that's what I mean. That's what it means. So other things, hydrogen extracellular fluid inside of the cell is negatively charged. So outside of the cell, this is something you're going to see with mitochondria, but he's talking about it here. I wish he would have talked about action potential a little bit more. Here's a sucrose molecules when they get inside. Of course, hydrogen ions get inside, and the hydrogen ions eventually have to go outside, and that is where ATP is needed. This is against gradient. There's less hydrogen here than here. So they are being pumped out because you need them. They are needed when sucrose is getting inside of the cell. You also need hydrogen to get inside of the cell as well. So diffusion of hydrogen. Okay, how bulk transport across plasma membrane occurs by exocytosis and endocytosis. We didn't talk about it in this class. I'm sorry. We didn't talk about it in this class. Small molecules of water enter and leave the cell through bilipid via transport. Proteins, aquaproteins, large molecules such as polysaccharides and proteins cross the membrane in bulk. They're huge. They cannot pass through the phospholipid bilayer. We talked about that. Bulk transport requires energy, exocytosis. It means exit the cell when they exit the cell, and endocytosis when they enter the cell, the materials, bulk molecules. Phagocytosis is formation of the vacuoles and cell eating. Okay, you will see it in a minute. Pinocytosis is cell drinking, and you will see it right here. Phagocytosis, big, huge molecule right here. It cannot go through the cell membrane easily. So what happens? A piece of cell membrane get pinched off. So this is cell membrane right here. And then of course forms a fluid vacuole. Pinocytosis, cell drinking. You have a lot of stuff in here. All of these are small molecules. Again, a piece of the cell membrane get pinched off. And then of course you have a vesicles. And then the third one is receptor-mediated endocytosis. Endocytosis is getting inside of the cell. So these are receptors, the Y-shaped receptors on the surface of the cell. This one did not have it. You guys watch. This one, it did not have it. But this one has these receptors. And then when these molecules, the green molecules, want to get inside of the cell, they have to attach, something has to attach to these receptors, and then the cell membrane will be pinched off. Here, there is no receptors involved. Okay, you don't see any receptors here, none. Okay, anyhow. What is receptor-mediated endocytosis? Very important concept. They teach receptor biology in major universities for one entire semester or quarter. So receptor-mediated endocytosis, finding of ligand. Ligand is any molecule like these, like the triangular. These triangular are called ligand, which they attach the receptors. This is not a ligand. The circles are not ligand because they're not attaching to any receptors. Okay, they just are in their free ride. How about that? That's the easy way to say it. However, so ligand receptor triggers, so just like lock and key. We use that terminology a lot in biology, lock and key, like enzymes and substrates, lock and key. So ligand and receptor is like lock and key. Okay. And when you open up the door, well, then a whole bunch of stuff get inside of the house too, right? But you actually have the key. Ligand is any molecule that binds specifically to a receptor. I'll talk about that. So here is showing you a, excuse me, here is showing you a transmission electron picture. This is a TEM. Okay, this is a TM picture showing you that a coat of protein is formed, and then these plasma membrane, let me see, receptor-mediated endocytosis. So the cell is being pinched off. The cell membrane is being pinched off right here, and the material goes inside the cell. And these are all ligand in here somewhere, protein codes inside of it, and so on and so forth. That's what this diagram is showing you. So, okay, so I'm all done with chapter 7, and I hope you guys enjoyed it. I'm gonna stop and then go ahead, go over chapter 11 later on as we come back to